All right, so here we're going to start talking about what sugars can do or how they're important in our body apart from energy. Um, an important aspect of sugar is to glycosylate. They can glycosylate lipids, they can glycosylate proteins, and it's all going to be very important in cell communication as well as cellular function. So we're going to start by talking about what exactly a glycoside is. We're going to talk about gluconarides. We're going to talk about the differences between our amino sugars, and we're going to talk about some specific sugars, cyalic acid, for example, and fructose. And then we're going to talk again about glycosaminoglycans and what a glyc is and how they're important. So a glycoside is basically consists of sugar bound to a different structure. And this different structure is a glyco, meaning that it's not another sugar. The sugars are going to attach themselves to from the hydroxyl group of the anomeric carbon. And you can be joined in various ways. We're going to have oxygen linkages, O-glycosides, sulfur linkages, nitrogen, and carbon linkages. So if we look at our glucose metabolism, so glucose is a sugar, of course, and we know glucose can go on to do many things. One of the things that we've been recently talking about is glucose can become glucose 1-phosphate, UDP glucose, and then become glycogen. And so here we're going to start talking about different pathways. We're going to talk about UDP gluconerate, or glycoproteins, glycolipids, and proteoglycans, which are the three types of molecules that can be glycosylated: proteins, lipids, glycans. And we also have UDP galactose, which we're going to talk about, which can become lactose. So basically, activated sugars, such as what we talked about with respect to building glycogen, UDP glucose, is where you attach nucleotides to the sugars, and then they get joining them to other things such as proteins and lipids. And this is all going to occur through those glycosidic bonds that we talked about when we talked about building up glycogen. Now this is an example of the different reactions of UDP glucose. Now UDP glucose is going to generate UDP gluconerate, which we'll talk about, and then we're going to talk about UDP galactose as well. And then in future lectures, we're going to talk about how we can make our glycoproteins and glycolipids. Now, UDP gluconerate is going to be negatively charged, and so it starts with UDP glucose, and then UDP glucose will become, through UDP glucose dehydrogenase, will reduce NAD and will oxidize your glucose basically to make UDP gluconerate. And this is going to enable it to bind to things to make them more soluble, such as drugs and xenobiotics. And so some group will come in, and then you're going to bind it to UDP gluconerate to make a gluconeride, which then can be excreted. So this is an important in getting rid of toxins in your body, for example, or things in your diet that you don't need or use. So here's an example of what UDP gluconerate can do. UDP gluconerate can bind to different xenobiotics, steroids, drugs, bilirubin, and become gluconerides and then excreted. It can be used to make bilirubin digluconeride, again get excreted. It's going to be able to generate proteoglycans, glycosylate proteins. It's going to be important in iuronidate, which is going to make, again, glycosaminoglycans, these GAGs. UDP xylose, again, important for glycosaminoglycans or making GAGs. Now, the formation of gluconerides, an example is bilirubin. Bilirubin is going to have negative charges attached to it. Again, it's going to help it to be able to be excreted. Bilirubin is a degradation product of heme. So your red blood cells are going to be broken down in your spleen, and then heme gets transported to the liver, bound to serum albumin. And then in the liver, you're going to add gluconerate residues through UDP gluconerate transfer to bilirubin. And this is also going to happen with many different types of drugs and steroids. It's going to be the same process. And then these gluconorides are going to, in the liver and the kidney, go through gluconeryl transferases, and you're going to basically just add these negative charges. Gluconerate itself can go through glucose metabolism through xylose 5-phosphate, which is one of those intermediates of the pentose phosphate pathway. So again, it's a way to get the carbons back into glucose metabolism as needed. Now, another thing that UDP glucose can do is generate UDP galactose. And so lactose is going to be synthesized from UDP galactose. So UDP glucose through that 4-epimerase is going to generate UDP galactose. And the formation of UDP galactose is going to be that first step that needs to be done in order to make lactose. Now, lactose synthesis is going to be important, of course, in women who are having children. And you're going to have a hormone, prolactin, that's going to lower the Km of galactosidyltransferase. And so this is going to allow the production of lactose from UDP galactose. If prolactin wasn't there, UDP galactose would not generate lactose, of course. It also requires another mole of glucose in order to become lactose, because remember, lactose is glucose and galactose. Alpha-lactalbumin glycosyl transfer activity, in the absence of that, you're not going to get lactose formed. Instead, your UDP galactose will form glycoproteins. It's also going to be used for glycosylation. Now, this, of course, is all synthesized into lactose synthase. Now we're going to talk about the interconversion of sugars. So we're going to have, for example, glycoproteins. Glycoproteins are proteins that are going to contain glycans, which are sugars. The carbohydrates are going to be part of a post-translation modification. We're going to have branches of carbohydrates chains that can be attached to asparagine, for example. And these are going to be transferred by dolichophosphate, and we'll talk about that in a future lecture. It's important for having glycoproteins. Glycoproteins are going to be important for interactions, cell to cell, cell to extracellular matrix. They're going to be on the cell membrane. By having the sugars added, it forms receptors, specificity for certain hormones, for example, or signaling. There's a group of sugars that are going to be used for both glycolipid and glycoprotein synthesis. And you can see here that we have all of these sugars are going to be activated. So we have a series of UDP activation. And then we have a CMP, which is very unique, and we'll talk about that. And then we have GDP. You're going to have enzymes that are going to be specific for both the sugar and the nucleotide. So the reactions of making the glycosylation is going to be specific. And then the sugar nucleotides are going to be essential building blocks for our proteins and our lipids, for them to function. So here is an image from your book about the interconversion of the sugars, and it looks very busy, but it's not that busy. So what we're basically talking about here is stuff we've already talked about. We have glucose, and glucose becomes glucose 6-phosphate. And then glucose 6-phosphate can become fructose 6-phosphate or glucose 1-phosphate. And then glucose 1-phosphate becomes UDP glucose, as we've just been talking about. And this, of course, can go on to form glycogen. But it can also become the stepping stone for glycosylating proteins and lipids. And so you can see here that we have iduronic acid, which is one of the building blocks of GAGs, glycosaminoglycans. We also have here UDP xylose, which can feed back into your pentose phosphate pathway. And this is glycogen. Now, an important thing is you're going to push fructose 6-phosphate to glucosamine 6-phosphate, and this is going to use glutamine. And so this step is critical for making many of the glucosamine, um, activated glucosamine in order to build many of our important products. So again, you can see here we have UDP glucosamine, which this can be important for heparin, for example. This is another GAG, Glycosaminoglycan. We also have UDP and acetylglucosamine, and then this can be UDP and acetylgalactosamine, and both of these are forming our GAGs again. And we also have, of course, once you have UDP and acetylglucosamine, this is going to form potentially our cyalic acid, and this is going to form that sugar, that CMP, which is very unique, and this is important for our gangliosides and glycoproteins. Now, we have our fructose. Maybe I can do this. Yeah. We have our fructose 6-phosphate all the way down to yes, it's our hexosamine. This red box here is hexosamine biosynthesis pathway. And so I'll be talking about this, but it's right here in this pathway from going from fructose 6-phosphate through UDP and acetylglucosamine. And so this pathway is becoming important as a pathway that is going to be a good sensor of glucose and glucose metabolism. So hexosamine biosynthesis pathway is going to be generated by a protein called Gfat, and that's shown here. Gfat is glucosamine fructose 6-phosphate amino transferase. And so you can see that it's sort of another byproduct of glycolysis. So remember, in glycolysis, we have glucose 6-phosphate, and this can go to the pentose phosphate pathway or glucose 1-phosphate to glycogen. Well, I'll put UDP glucose. But fructose 6-phosphate through Gfat can become glucosamine 6-phosphate. And overexpression of Gfat leads to what we refer to as peripheral insulin resistance. And this is especially important, it turns out, to be in adipose tissue. So one thing that's happening with Gfat is you're generating an acetylglucosamine down at the very end. And this is going to be used to phosphorylate, or glycosylate, sorry, not phosphorylate, glycosylate. And this is going to be linked through oxygen, through the O, and this is going to target many of those enzymes that are involved in insulin. So our insulin receptor substrate 1 will get glycosylated. Phosphofructokinase, phosphoinositide 3 kinase will get phosphorylated, etc. Also, proteins involved in translocation of GLUT4, and so in our adipose tissue we have GLUT4, and we need to get GLUT4 to the surface. And it's thought that overexpression of Gfat pushing things through this is going to alter glycosylation. You're going to get too much of your N-acetylglucosamine, and then it's going to inhibit, for example, your GLUT4 receptor from being able to be expressed on adipose tissue in the presence of insulin and glucose. This is all being still under investigation, and so this, but it's well known now that this pathway is important in type 2 diabetes. Now, another thing that's important, though, you do need to push through UDP and acetylglucosamine because you need to generate your glycosaminoglycans. Your glycosaminoglycans are very, very important in extracellular matrix, also important on the surface of cells. They have a lot of negative charges. They interact with water. They are going to be very important in cushioning. They're going to be very important in the viscosity of your extracellular matrix. They're either going to be made up of a UDP N-acetylglucosamine or N-acetylgalactosamine, and a uronic acid such as gluconerate or iduronate. Some examples of your GAGs that are very important are hyaluronic acid, dermatan sulfate, chondroitin sulfate, heparin, heparin sulfate, and keratin sulfate. Some of the things to keep in mind is your hyaluronic acid does not contain sulfate. So this is very important. It's very unique about this. It's going to be important in lubrication and shock absorber. So you're going to find this in extracellular matrix, subconnective tissue, cartilage, etc. Dermatan sulfate is going to be important for coagulation and wound healing, infections, etc. It can have some excess accumulation, can have some phenotypes as well. We're going to have the most abundant glycosaminoglycan is chondroitin, which is going to be our proteoglycans, and this is going to be important in cartilage, bone, heart valves. Heparin is going to be anticoagulant. We have heparin sulfate is going to have many functions, and one of the functions it has is also a receptor, or it's going to be a ligand for chylomicron remnants. And so it's going to enable the liver to pick up chylomicron remnants. Keratin sulfate is going to be proteoglycans also in cornea, bone, and cartilage. Another odd sugar that is part of this pathway is our fructose. So our fructose is going to be found in N-linked glycans. So we're going to have a lack of our hydroxyl group at that position 6 on our fructose. And fructose is rare in that it has the L conformation. Another thing to point out about the interconversion is the amino sugars, including what we were just talking about, N-acetylglucosamine, is going to all come from glucosamine 6-phosphate. So we're going to have fructose 6-phosphate, again, through glutamine, a transfer of that nitrogen, and you're going to get glucosamine 6-phosphate. So all amino sugars are derived from this glucosamine 6-phosphate. And then you're going to have an acetylation to create an acetylglucosamine 6-phosphate, which is going to be a precursor for your glycosaminoglycans in particular, as well as a precursor for sialic acid. So the importance here is glucosamine. It can be N-acetylated by the acetyltransferases. This is going to be in our endoplasmic reticulum as well as the cytosol. This can also be a way to modify sugars, drugs, xenobiotic compounds, another mechanism to add negative groups so that you can excrete those. Now, sialic acid or NANA and acetyluramic acid is going to be made from N-acetylmanosamine. So again, we have our glucosamine 6-phosphate gets acetylated, and after acetylation, it has 2 routes to become sialic acid. It can go through UDP and acetylglucosamine, or it can go through N-acetylglucosamine without the UDP, and it becomes basically manosamine. So N-acetylmanosamine is a precursor for sialic acid. Now, the other things that are pretty interesting about sialic acid is not only is it important because it adds negative charges to proteins exterior of our cells so that can interact with the aqueous environment, and also it's going to be very important in communicating cell-to-cell communication. But it's going to, its production is going to start in the cytoplasm, but it has to go to the nucleus to get activated. The enzyme to activate sialic acid is going to be in the nucleus, and it's by an N-acetylneuraminate CMP pyrophosphorylate. And basically, you're going to take your Cyalic acid. So I guess I can do it here. You have, I'm going to put this up second, right? You have ATP. Oops. We're going to have CTP, sorry. And then you're going to get PPI released, and then you're going to get that CMP attached to your N-acetyluramic acid, and that is the active sialic acid. And then active sialic acid is going to be important in gangliosides, for example, as well as it can be glycosylate proteins too. And we'll talk more about that when we talk about lipids. Now, the other thing to point out about NANA, it's the only sugar in our metabolism that has a carrier monophosphate, that CMP. It's the only one that has that. Everything else is going to be a di, UDP, etc. So let's just talk a little bit about what glycosylation is. Glycosylation basically is a sugar is transferred from the nucleotide sugar, which is an activated sugar, to some other group. And in this case, we're attaching it to a protein. So the activated sugar is going to provide the energy necessary for the transfer. And the enzymes are going to be transferases. Again, they're going to be specific for the sugar as well as the nucleotide attached to it. And so a glycosyltransferase is going to transfer sugars from the nucleotide sugar to a nucleophilic amino acid on proteins, or it could be a hydroxyl group or of another sugar. But here again, we're doing a glycosylated protein.